अस्सलाम वालेकुम स्टूडेंट्स। होप यू all are fine। स्टूडेंट्स आज हम बायो क्लास 11 चैप्टर नंबर 4 एक्सरसाइज शॉर्ट क्वेश्चन आंसर्स सॉल्व करेंगे। सो लेट्स स्टार्ट। फर्स्ट क्वेश्चन है हमारे पास, व्हाट इज इलेक्ट्रोमैग्नेटिक स्पेक्ट्रम? आंसर इज the electromagnetic spectrum is the range of all possible frequencies of electromagnetic radiation, which includes radio waves, longest wavelength, lowest frequency, microwaves, infrared, IR radiation, heat, visible light, what we can see, ultraviolet UV radiation, X-rays, gamma rays, shortest wavelength, highest frequency. These types of radiation differ in their wavelength, distance between peaks, frequency, number of peaks per second, energy. Higher frequency means higher energy. The electromagnetic spectrum is a continuous range with no clear boundaries between types. It's a fundamental concept in physics crucial for understanding various phenomena from communication technology to medical imaging and astronomy. Here's a rough ordering of the spectrum from longest wavelength to shortest: radio waves, then microwaves, then infrared radiation, visible light, ultraviolet radiation, X-rays, and gamma rays. Next question is, explain action spectrum of photosynthesis. The action spectrum of photosynthesis is a graphical representation of the rate of photosynthesis versus the wavelength of light. It shows which wavelength of light are most effective at driving photosynthesis. In other words, it measures how efficiently plants use different colors of light to produce glucose and oxygen. Here's a key takeaway: blue light 400 to 450 nanometer and red light 600 to 700 nanometer are most effective for photosynthesis. Green light 500 to 550 nanometer is least effective as plants reflect this wavelength, which is why they appear green. This spectrum is important because it helps us understand how plants adapt to different light environments, informs strategies for optimizing plant growth in agricultural settings, has implications for the design of artificial lighting systems for plant growth. The action spectrum is typically plotted as a curve with photosynthetic rate on the y-axis and wavelength on the x-axis. This curve shows a double-peaked shape with peaks in the blue and red regions. Next question is, what are the types of chlorophyll? There are several types of chlorophyll, but the main ones are chlorophyll A. This is the most important and abundant form responsible for absorbing blue and red light. It's present in all photosynthetic organisms. Chlorophyll B. This form absorbs blue, violet, and red light and is present in plants, algae, and cyanobacteria. Chlorophyll C, found in some algae and phytoplankton, it absorbs blue, violet, and red light. Chlorophyll D, present in some red algae, it absorbs blue and red light. Chlorophyll F, recently discovered, it absorbs far red light and is present in some cyanobacteria. These types differ in their molecular structure, absorption spectra, wavelengths of light absorbed, functions in photosynthesis. Chlorophyll A is the primary pigment, while others play accessory roles, transferring energy to chlorophyll A. This teamwork enables efficient photosynthesis across various light environments. Next is, what is importance of carotene? Carotene is a yellow-orange pigment found in plants, algae, and photosynthetic bacteria. Its importance includes antioxidant properties. Carotene protects cells from damage caused by free radicals, which can lead to chronic diseases like cancer and heart disease. Precursor to vitamin A. Carotene is converted to vitamin A in the body, essential for vision, immune function, and skin health. Photosynthesis. Carotene plays a role in photosynthesis, transferring energy to chlorophyll and protecting plants from excessive light. Coloration. Carotene is responsible for yellow, orange, and red colors of many fruits and vegetables. Health benefits. A diet rich in carotene has been linked to reduced risk of age-related muscular degeneration, cataracts, certain cancers, for example lung, breast and colon, cardiovascular disease. Common sources of carotene include leafy greens, kale, spinach, orange and yellow fruits like carrots, sweet potatoes, pumpkins, tomatoes, squash, peppers. The next is describe absorption spectrum in photosynthesis. The absorption spectrum in photosynthesis refers to the range of wavelengths, colors of light that are absorbed by pigments such as chlorophyll and other accessory pigments in photosynthetic organisms. Here's a breakdown. Chlorophyll A absorbs blue light 430 to 450 nanometer, absorbs red light 650 to 700 nanometer, reflects green light 500 to 550 nanometer, which is why plants appear green. Chlorophyll B absorbs blue-violet light 450 to 495 nanometer, absorbs red light 600 to 650 nanometer, accessory pigments, for example carotene, phycobiloproteins, absorb light in the blue, violet and red regions, transfer energy to chlorophyll A. The absorption spectrum shows peaks, wavelengths with maximum absorption, troughs, wavelengths with minimal absorption. This spectrum is important because it determines the types of light that can be used for photosynthesis, influences the rate of photosynthesis, helps plants adapt to different light environments. Keep in mind that the absorption spectrum can vary slightly between species and environmental conditions. Next question is, what is photosystem? Explain. A photosystem is a complex of proteins and pigments found in the thalloid membranes of chloroplasts responsible for converting light energy into chemical energy during photosynthesis. There are two main photosystems. Photosystem 1 absorbs light with a wavelength of 700 nanometer. P700 generates a high energy electron pair used in cyclic electron flow to produce ATP and NADPH. Photosystem 2 absorbs light with a wavelength of 680 nanometer. P680 generates a high-energy electron pair used in non-cyclic electron flow to produce ATP, NADPH, and oxygen. Each photosystem consists of reaction center, where light energy is converted into chemical energy, antenna pigments absorb light energy and transfer it to the reaction center. Electron acceptors receive electrons from the reaction center and pass them along the electron transport chain. Photosystems work together to generate ATP and NADPH produce oxygen through water oxidation, drive the electron transport chain. In summary, photosystems are the functional units of photosynthesis, harnessing light energy to power the conversion of carbon dioxide and water into glucose and oxygen. Next question is, what is the role of carbon dioxide in photosynthesis? Carbon dioxide plays a crucial role in photosynthesis, serving as the primary source of carbon for glucose production. Here's its role. Carbon source. Carbon dioxide provides carbon atoms necessary for synthesizing glucose. C6H12O6 accepts electrons. Carbon dioxide accepts electrons from the reduced coenzymes NADPH and ATP produced in the light-dependent reactions. Reduction. Carbon dioxide is reduced to form organic compounds, releasing oxygen as a byproduct. Fixation. Carbon dioxide is fixed into a 3-carbon molecule called 3-phosphoglycerate 3-PGA via the enzyme Rubisco. Energy storage. The energy from light is stored in the bonds of glucose, which is synthesized from carbon dioxide. In summary, carbon dioxide is the carbon source for glucose production, accepting electrons and being reduced to form organic compounds, releasing oxygen and storing energy in glucose bonds. Here's the simplified equation. 6CO2 plus 6H2O, 6 molecules of carbon dioxide plus 6 molecules of water plus night energy gives us C6H12O6 glucose plus 6 molecules of oxygen O2. Next question is, how it was confirmed that plants split water as a source of hydrogen, releasing oxygen as byproduct. The discovery of water splitting in photosynthesis was a gradual process involving several scientists and experiments. Here's a brief overview. First one is Jan Ingenhousz, 1779, observed that plants release bubbles when placed in sunlight, suggesting gas production. Next is Jean Senibeer, 1782, demonstrated that the gas released is oxygen and its production is proportional to light intensity. Next is Nicholas Theodore, 1804, showed that plants' growth requires both carbon dioxide and water, implying water's role in photosynthesis. Next is Cornelis van Niel, 1931, proposed the concept of water splitting, suggesting that light energy is used to split water into hydrogen and oxygen. Samuel Rubin and Martin Kamen, 1941, used isotopic labeling deuterium oxide (D2O) to demonstrate water splitting, showing that oxygen is produced from water, not carbon dioxide. Hill reaction, 1937. Robert Hill discovered that isolated chloroplasts can produce oxygen when illuminated, confirming water splitting. Kelvin cycle, 1950s. Melvin Kelvin and colleagues elucidated the light-independent reactions, further supporting water splitting. These experiments collectively confirmed that plants split water as a source of hydrogen, releasing oxygen as a byproduct during photosynthesis. Next is, what is importance of G3P? G3P stands for glyceraldehyde 3-phosphate, is a crucial molecule in photosynthesis and cellular metabolism. Its importance includes energy storage. G3P is a high energy intermediate storing energy from light absorption during photosynthesis. Glucose synthesis. G3P is a building block for glucose C6H12O6 synthesis, combining with another G3P molecule to form fructose 1,6-bisphosphate. Redox reactions. G3P is involved in redox reactions, regenerating NADPH and ATP in the Calvin cycle. Cellular metabolism. G3P is a key intermediate in glycolysis, gluconeogenesis, and the pentose phosphate pathway. Reducing power. G3P provides reducing power for various cellular processes including fatty acid synthesis and amino acid production. Carbon skeleton. G3P serves as a carbon skeleton for the synthesis of other organic compounds. In summary, G3P is a critical molecule that stores energy from light, contributes to glucose synthesis, participates in redox reactions, plays a role in various metabolic pathways, provides reducing power, serves as a carbon skeleton. Its importance extends beyond photosynthesis, impacting our cellular metabolism and energy production. Next question is, what is the effect of temperature on the activities of rubisco? Temperature significantly affects the activity of rubisco ribulose 1,5-bisphosphate carboxylase oxygenase, the enzyme responsible for fixing carbon dioxide in photosynthesis. Here's how temperature impacts rubisco activity. Optimum temperature. Rubisco has an optimal temperature range of 25 to 30 degrees Celsius where its activity is highest. Next is temperature increase. Above the optimum temperature 30 to 40 degrees Celsius, rubisco activity decreases due to enzyme denaturation, reduced substrate binding, increased oxygenase activity leading to photorespiration. Temperature decrease. Below the optimum temperature 15 to 25 degrees Celsius, rubisco activity also decreases due to reduced enzyme flexibility, slower substrate binding, increased inhibition by oxygen, high temperatures. Prolonged exposure to high temperatures greater than 40 degrees Celsius can lead to enzyme degradation, loss of activity, reduced photosynthetic capacity. Low temperatures. Chilling temperatures less than 15 degrees Celsius can slow down riboscope activity, increase the affinity for oxygen, leading to photorespiration. Temperature affects riboscope activity by altering enzyme structure, substrate binding, and reaction rates. Understanding these effects is crucial for optimizing photosynthetic performance in various environmental conditions. Next question is, what are the disadvantages of photorespiration? Photorespiration is a process in plants that occurs when rubisco, the enzyme responsible for fixing carbon dioxide, also reacts with oxygen, leading to energy loss and reduced photosynthetic efficiency. The disadvantages of photorespiration include reduced photosynthetic rate. Photorespiration decreases the rate of photosynthesis, leading to reduced plant growth and productivity. Energy loss. Photorespiration consumes energy ATP and NADPH generated during light-dependent reactions, reducing the energy available for carbon fixation. Carbon loss. Photorespiration leads to the release of carbon dioxide, reducing the amount of carbon available for glucose synthesis. Oxygen consumption. Photorespiration consumes oxygen which could otherwise be used for other cellular processes. Increased water usage. Photorespiration can lead to increased water loss through transpiration as plants try to compensate for reduced photosynthetic activity. Reduced water use efficiency. Photorespiration reduces the amount of carbon fixed per unit of water used, decreasing water use efficiency. Impacts on plant productivity. Photorespiration can limit plant productivity, particularly in hot and dry environments, where energy and water are already limited. Increased oxidative stress. Photorespiration can lead to the formation of reactive oxygen species (ROS), causing oxidative stress and damage to plant cells. Overall, photorespiration reduces photosynthetic efficiency, plant growth, and productivity, making it a significant disadvantage for plants. Next question is, how photorespiration evolved? Photorespiration is believed to have evolved as a consequence of the oxygenation of the Earth's atmosphere, which occurred around 2.7 billion years ago. Here's a simplified overview of its evolution. Early Earth atmosphere. The atmosphere was mostly devoid of oxygen, and photosynthesis was likely anoxygenic, not producing oxygen. Oxygenation of the atmosphere. As cyanobacteria evolved and became dominant, they produced oxygen through oxygenic photosynthesis, gradually oxygenating the atmosphere. Rubisco evolution. The enzyme Rubisco, responsible for fixing carbon dioxide, evolved to accommodate the changing atmosphere. However, its active site was not optimized for the new oxygen-rich environment. Oxygen binding. Rubisco's active site began binding oxygen instead of carbon dioxide, leading to the formation of phosphoglycolate and the initiation of photorespiration. Photorespiratory pathway evolution. The photorespiratory pathway evolved to recycle phosphoglycolate and recover some of the energy lost during photorespiration. Adaptation and optimization. Over time, plants adapted to photorespiration by optimizing the photorespiratory pathway, developing mechanisms to reduce its impact, and evolving new strategies to conserve energy. Photorespiration is now an integral part of the photosynthetic process, and this evolution has shaped the development of plant metabolism and ecology. Next question is, write differences between chlorophyll A and chlorophyll B, carotene and xanthophyll, action spectrum and absorption spectrum of chlorophyll A and chlorophyll B, antenna complex and reaction center, photosystem 1 and photosystem 2, light-dependent reactions and light-independent reactions of photosynthesis, oxidative phosphorylation and photophosphorylation, cyclic photophosphorylation and non-cyclic photophosphorylation, C4 carbon fixation and C3 carbon fixation, lactic acid fermentation and alcoholic fermentation, Calvin cycle and Krebs cycle, oxidative phosphorylation and substrate level phosphorylation. So first of all, difference between chlorophyll A and chlorophyll B. Chlorophyll A absorbs blue and red light, reflects green light, and is the primary pigment for photosynthesis. Chlorophyll B absorbs blue violet and red light, reflects yellow green light, and is an accessory pigment. Carotene and xanthophyll. Carotene, yellow orange pigment, absorbs blue, violet and red light and is responsible for yellow and orange colors. Xanthophyll, yellow pigment, absorbs blue violet light and is responsible for yellow colors. Action spectrum and absorption spectrum. Action spectrum measures the effectiveness of light in driving photosynthesis. Absorption spectrum measures the absorption of light by pigments. Next is absorption spectrum of chlorophyll A and chlorophyll B. Chlorophyll A absorbs light at 430 nanometer and 660 nanometer. Chlorophyll B absorbs light at 450 nanometer and 640 nanometer. Antenna complex and reaction center. Antenna complex collects light energy and transfers it to the reaction center. Reaction center converts light energy into chemical energy. Photosystem 1 and Photosystem 2. Photosystem 1 absorbs light at 700 nanometer, generates high energy electrons and produces NADPH. Photosystem 2 absorbs light at 680 nanometer, generates high energy electrons and produces ATP and oxygen. Light dependent reactions and light independent reactions. Light dependent reactions. Light depend occurs in thalakite, require light and produce ATP and NADPH. Light independent reactions or Calvin cycle occur in stroma, don't require light and produce glucose. Oxidative phosphorylation and photophosphorylation. Oxidative phosphorylation generates ATP in mitochondria through electron transport chain. Photophosphorylation generates ATP in chloroplast through light energy. Cyclic photophosphorylation and non-cyclic photophosphorylation. Cyclic photophosphorylation produces ATP only, involves photosystem 1. Non-cyclic photophosphorylation produces ATP and NADPH, involves both photosystems. C4 carbon fixation and C3 carbon fixation. C4 carbon fixation. Adapted to high temperature and low carbon dioxide, uses PEP carboxylase. C3 carbon fixation uses Rubisco, more common and efficient. Lactic acid fermentation and alcoholic fermentation. Lactic acid fermentation occurs in mussels, produces lactic acid. Alcoholic fermentation occurs in yeast, produces ethanol and carbon dioxide. Calvin cycle and Krebs cycle. Calvin cycle occurs in chloroplast, produces glucose from carbon dioxide. Krebs cycle, also called citric acid cycle, occurs in mitochondria, produces ATP, NADH and FADH2. Oxidative phosphorylation and substrate level phosphorylation. Oxidative phosphorylation generate ATP through electron transport chain. Substrate level phosphorylation generates ATP through direct energy transfer from substrate. So students, हमारे क्वेश्चंस कंप्लीट हो चुके हैं. होपफुली आपको समझ आ गए होंगे. If you have any doubt, you can ask in comment section. Please like, share and subscribe my YouTube channel. Thank you.